Thank you so much for having me here today. I'd like to take this opportunity to especially thank the DYSS organizers for having me here as one of these speakers. I'm very excited to share some of my doctoral and postdoctoral research on grafted Zwitterionic polymers with biomolecules at nanoscale. So as it's important. Okay, thank you. As Brendan mentioned, I did my PhD with Professor Deborah Leckland in the University of Illinois at Urbana-Champaign in the Department of Chemical and Biomolecular Engineering. So in my first project, I looked at how protein interacts with grafted Zwitterionic polymers at various grafting densities as well as ionic strength of the surrounding medium. The results from this study helped reconcile what we see in solution with Zwitterionic polymers as opposed to a very non-fouling properties that are commonly observed for grafted Zwitterionic polymers. And in the second project, I measured surface forces of grafted Zwitterionic polymers at varying ionic strength and grafting densities. And this results from this project help to understand what are the magnitude as well as ranges of distances that are important for developing nanoparticle stabilizers, for example. And then in the third project, I will talk about surface forces of statistical copolymers at varying Zwitterionic composition. And in my postdoctoral research, I'm doing currently research with Professor Roberto Henderson-Inculis in the Department of Material Science and Engineering. And here I'm looking at modeling surface interactions happening at the articular cartilage surface with synovial fluid components. And particularly, I'm focusing on the role of fibronectin and how it interacts in comparison with collagen type 2 with the different components from synovial fluid, particularly hyaluronic acid and lubricin. So with in the future work, I want to focus on grafted Zwitterionic polymer brush for developing better lubricating load-bearing thin films. And so here the goal is to understand absorption and retention of synovial fluid as well as the hydration, lubrication properties of Zwitterionic polymers. With that, first, as a lot of you know that Zwitterionic polymers are very interesting polymers. They have both positive and negatively charged moieties in their side chains. And although they are locally charged, but they are overall charge neutral. And these are the three of the most commonly studied repeat units of Zwitterionic polymers. And in my research, I focused on sulfobutane methyl acrylate, consisting of positively charged quaternary ammonium ion and sulfonate ion as the negatively charged moiety in the side chain. So we care about studying Zwitterionic polymers, especially to modify surfaces, because it shows very low non-specific protein adsorption, or otherwise known as fouling. And they're even categorized as ultra-low fouling, which means that for blood contacting devices, it has been shown that by modifying the surfaces with grafted Zwitterionic polymer, it leads to less than 5 nanogram per centimeter square fibrinogen adsorption, which is the gold standard for avoiding the body's immune response. And in recent study, it has been shown that Zwitterionic polymers do interact with proteins in solution. And it was shown that the positive and negatively charged ions and their spacing, which is shown here as red and blue balls along the yellow polymer backbone, they are found to be very important in interacting with the charged patches on the surface of the protein. And so the question that I wanted to address is, why do polysulfobutanes, as a model, Zwitterionic polymers, they interact with proteins in solution, but they show really good repelling properties when grafted on substrate? So the hypothesis we wanted to test is that the grafted polysulfobutanes behave as weakly attractive grafted chains to proteins or colloids. And before I explain what that is, I wanted to briefly mention the 3 modes of protein adsorption for grafted polymer chains on substrate. So in scenario A, when the protein is small enough that it can penetrate the brush layer, then it can adsorb on the underlying substrate, which is known as the primary adsorption. In scenario C, when the protein is too big, that the insertion penalty into the brush layer is very high for the protein to overcome, it can adsorb on the outer layer of the brush, which is known as secondary adsorption. And in an intermediate scenario, the protein is small enough that it can penetrate into the brush layer, overcoming the osmotic penalty. And then they start forming this attractive segment protein interactions, which allow it to adsorb on the grafted chains. So for this scenario, Halperin and Kroger, in this paper, they developed a theoretical model that showed that if you plot the normalized surface density of adsorbed protein as a function of area per chain or the grafting density, then the adsorption profile is bell-shaped rather than monotonically decreasing. So what happens is that at both ends, when you have a highly dense brush or a isolated mushroom-like coil chain, the adsorption is low, but at an intermediate surface density, which corresponds to a weakly overlapping chain, the protein adsorption is the highest. And so we wanted to see that for grafted polysulfobutanes, if we change the polymer configuration on the substrate and change the grafting density, can we see this type of bell-shaped curve for different types of proteins? So in order to do that, quite simply, we synthesize the polysulfobutane chains in the graft from method by using atom transfer radical polymerization, or ATRP, which also allowed us to control the polydispersity as well as have a good control over the molecular weight of the synthesized brush. And then we measured different types of protein solution using surface plasmon resonance and ellipsometry. So the goal was to understand the protein adsorption mechanism. So, so by varying the OH terminated thiol and bromine terminated thiol, bromine being the active part of the initiator, in ATRP equilibrium, we can regulate the distances between the polymer chains, which controls the grafting density. And we can also change the chain length or the molecular weight of the polymer. And from that, we can estimate the Flory radius. And the ratio, we use the ratio of S/2RF in order to define the polymer configuration. So when the ratio is much bigger than 1, then you get non-overlapping isolated mushroom-like chains on the substrate. And when the ratio is much less than 1, then you have densely grafted brushes. So then we did the synthesis by using a linear dipper. We took the gold-coated silicon wafer to graft a OH terminated thiol gradient along the dipping direction, and then backfilled with a bromine terminated thiol, and then performed the ATRP reaction in a custom-made reaction vessel in an inert condition to grow the polysulfobutane gradient on the substrate. And then we characterize the surface by using ellipsometry to measure the dry thickness of the grafted film. And we see a nice variation along the dipping direction. And we also use the X-ray-photoelectron spectroscopy to measure the chemical composition of the thin film. And from bromine to sulfur ratio, we estimated the the distances between the grafted chain, which is the inverse squared of the grafting density. So, and from the thin film dry thickness, we can estimate the molecular weight to determine finally the S/2RF ratio. And there's lots of things going on, but the idea is to graft different polymer configuration on the same substrate. And it ranged from brush to weakly overlapping to mushroom-like chain. And then we performed, then we monitored protein adsorption. So here, the graph on the right is showing the lysozyme adsorption as a function of S/2RF ratio. So as we go from left to right, the grafting density decreases. And we see that the adsorption profile is bell-shaped, and the amount of protein adsorption dependent on the ionic strength. So as we increase the salt concentration of the protein solution, the adsorption, the S/2RF, sorry, the adsorption maxima went down. So this means that the in water, which is the poor solvent for the polymers, the coils are shrunken and the protein can penetrate easily through the layer and adsorb on the polymer chain. However, as more and more salt ions are present, as we increase the ionic strength of the solution, there is now competition for the ions with the polar or the charged sides of the side chain of the polymer. And we see very low adsorption for when the density of the brush is very high, which represents the scenario of the ultra low fouling properties of polysulfobetanes that has been reported in many previous literature. So with that, there's just some other example of the proteins that we looked at. PGK is the phosphoglycerate kinase, a small metabolite protein, and we see the similar bell-shaped curve, and we also see the similar protein adsorption profile for fetal bovine serum as well as bovine serum albumin. So this is the signature of colloid interactions with weakly attractive grafted chains. And so from theory now we can describe the protein adsorption mechanism. And the underlying physics is the following. When there is a highly dense brush grafted on the substrate, the insertion penalty for the protein is too high. So therefore we see low fouling or low protein adsorption. But as the distances between the two neighboring chain increases, that means the grafting density decreases. Now the protein can overcome this osmotic repulsion by interacting with the surrounding polymer chain, which is attractive interactions. And so with lower grafting density, now the interactions increases, and so the amount of protein adsorption increases, reaches the maximum, and then it further goes down because here there is not enough polymer chains on the surface to interact with. And finally, at point E, the adsorption profile plateaus at an amount that is expected on a control substrate, which is OH terminator tile for our case. Then moving on to the second part of the project, I was interested in how the ionic strength dependent swelling behavior affect the surface forces of grafted Zwitterionic polymers. And the motivation for this project is that Zwitterionic polymers very effectively can stabilize nanoparticles in high temperature and high salt concentrated solution. Polyelectrolytes are not an ideal choice because, the electrostatics with salt present in the surrounding solution, there is electrostatic screening of the charges of the side chains of the charged polymers. And so that leads to a collapse of the chain, and there is loss of steric stabilization. So the nanoparticles would aggregate. In this work, using a small amount of polysulfobutanes to decorate the surfaces of iron oxide nanoparticles, it was shown that they can be sterically and electrostatically stabilized. So if we draw a distance, the interaction energy of the particles as a function of distance, then we see that it is a purely repulsive curve. With decreasing distance, the interaction energy increases. So surface forces are very important, and it determines how effectively the repulsive barriers will be generated by polymer-coated substrates. So the research question here is that we asked or addressed is, how does ionic strength-dependent swelling behavior influence the surface forces of thin polysulfobutane films? In solution, it is known that Zwitterionic polymers swell or increase in size with increasing salt. So if you measure the hydrodynamic radius with dynamic light scattering and plot it with increasing salt solution, then we see that at lower salt concentration, there are huge aggregates that are formed because of excessive self-association or the interactions between the side chains from the same polymer coil or with neighboring polymer coils. And as we add more and more salt, the coil dissolves and starts to interact with the charged side chains and expands. And so here, after reaching a minima, it expands further, and finally there is and we can see it visually also. And the coil dimension eventually becomes independent of increasing salt because there's now a chemical equilibrium that is established. So in this project, we wanted to see how the range and amplitude of the surface forces are affected by the swelling dependent behavior of the polysulfobutane chains. And in order to do that, we used a surface force apparatus. So just briefly, how it works is that there is a white light that passes through to reflecting surfaces, and the reflecting surfaces are decorated with the molecules of our interest. So in our case, it was a gold-coated hemicylindrical silica surface on the one side, and we grafted different configuration of polysulfobutane on gold. And on the other side, we have a back silver mica. And then after light interference happens, the light is passed through a spectrometer, which allows us to see the interference fringes, and we can measure the force from the deflection of the two interacting surfaces. And by applying the Riemann approximation, we can normalize the surface force in order to get the interaction energy. So here, apart from measuring the magnitude and range of surface forces, we were also interested in quantitatively compare between comparing the force distance curves at different ionic strength. And so we used a theory of simple polymer chains for quantitative comparison. So just briefly speaking, we worked with two different grafting density of polymers, brush and mushroom-like polymer chains. And let's take a look at the surface forces at high grafting density or brush-like polymer chain on the surface. And so on this graph, normalized surface force is plotted against separation distance. And we see that the onset of repulsion takes place around 50 nanometer, and it's a purely repulsive curve. So there is a decrease with the decreasing distance, the interaction forces increase, and the onset of repulsion is further pushed back further at 80 nanometer, and it remains about the same for even higher ionic strength or the salt concentration of the surrounding solution. And in order to compare quantitatively these three force distance curve, we fitted this force distance profile to Miller-Witten-Cates theory, and which describes the interaction force as a function of chain extension and distance. And we also fitted the prefactor, which scales with the surface force. And we showed that the chain extension, the chain extension increased by twofold as we go from 54 to 144 millimolar, as well as 1044 millimolar salt concentration. And when we compared the two higher salt concentration, we see that the range of repulsive barrier remains the same, but the surface forces that were measured were higher. So the brushes were less compressible in the highest ionic strength. Then we also looked at surface forces at low grafting density, and the force distance profiles for the three salt concentration are plotted on the same graph. And here we see a qualitatively different behavior. So at the smaller separation distance, the surface force dependence on the distance decreased exponentially, and we can fit the exponential behavior and determine the decay length, which gives us the effective radius of the polymer chain. And this increases with the salt concentration. And however, the prefactor remains the same because it's a function of the grafting density, and we have now changed the grafting density in these measurements. So with that, we looked at the different ranges of distances as well as the magnitude of the repulsive barrier generated by both high and low grafting density of polysulfobutane chains. And we showed that even at low grafting density or mushroom-like chains, the repulsive force can be substantial. So they can also have potential application for stabilizing nanoparticles. And also understanding that the effectiveness of steric barrier in electrolyte solution can be affected by the ionic strength dependent swelling behavior of the Zwitterionic polymers. So in the next project, I started investigating the surface forces of statistical copolymer with a non-ionic component, which is the oligoethylene methyl acrylate, and a Zwitterionic content, which is the sulfobutane methyl acrylate, in a statistical copolymer, and see how the surface forces are influenced by the different different grafting density and ionic strength of the polymer. And the biggest motivation for that is that statistical copolymers are great for preparing chemically heterogeneous materials. And for example, in this work done by Dan Schwartz's group, sulfobutane and oligoethylene methacrylate based polymer, statistical copolymer, was used to stabilize fibronectin. And it was shown that at an intermediate Zwitterionic content, most stability of the protein was achieved. And so we wanted to follow up with that. And for that particular statistical copolymer, we wanted to understand how do the surface forces vary with copolymer composition, and do the copolymers behave as simple mixture of independent components, or do they phase aggregate that alters the thin film structure? So we use we use the surface force apparatus to measure forces of the copolymer chain in one surface and mica as our testing surface. And we varied the relative amount of sulfobutane and oligoethylene methyl acrylate monomer to get different composition of the chain. And we characterized the brush composition by doing reflective FTIR spectroscopy. And we decided to work with only 0, 40, and 80% sulfobutane content. And we also had both brush. We looked at brush with a S/2RF ratio of 0.1 and weakly overlapping chains having a ratio of S/2RF around approximately 0.4. So first, with PEG-only polymer chains with no Zwitterionic component, we see that the force distance profile for both high and low density polymers, they overlap on each other. So there is no ionic strength dependent behavior, and that's not surprising. On the other hand, if you look at the 40% sulfobutane content in the statistical copolymer, we see that both the range of forces as well as the surface force measured at a certain separation distance increases. And this difference is more apparent when you have 80% go up to 80% sulfobutane content in the copolymer. And again, for quantitative comparison, we fitted this data with Milner, Witt, and Cates theory, which is more appropriate for high density polymer brushes. And we see that the chain extension, or L0, increases with salt concentration, and we see an increase in chain length only for the 1044 millimolar condition when we increase the sulfobutane content in the copolymer chain. So that means that using just varying just the monomer composition, we can get a proportional change in the chain extension and the surface forces. And when we looked at the surface forces of weakly overlapping copolymer chains with the S/2RF ratio of approximately 0.4, we see that the force distance profile is qualitatively similar to that of isolated mushroom-like chains. And we see an increase in effective radius of the chain with an increasing ionic strength. And this was also seen for 80% sulfobutane content. And therefore, just quantitatively showing you that the effective radius of the polymer chain increases with salt concentration as well as percentage sulfobutane content of the copolymer. With that, we showed that there's a proportional chain in equilibrium chain extension, coil size, as well as corresponding surface forces with percentage change in the sulfobutane content in copolymer chains. And we didn't measure any adhesion, or there wasn't any evidence of phase aggregation. And so we concluded that the components behave as ideal miscible monomers, and they're not interacting. And so they are contributing independently to the chain extension and chain interactions with ions. So now I would like to shift gears and talk a little bit about modeling articular cartilage. So in this current postdoctoral research, I am looking at how to mimic the surface interactions that take place in articular cartilage, where you have cartilage proteins such as fibronectin and collagen type 2, and it helps forming a thin lubricating layer from synovial fluid components, which consist of different types of aggregants, lubricin, hyaluronic acid. And this thin film is very interesting mechanical properties. It has some interesting mechanical properties because it helps to dissipate energy and also facilitates proper joint movement, and it decreases the coefficient of friction, all of which are very important for articulation of the bones. So we want to see in this project what is the role of fibronectin in comparison with collagen type 2 in forming the synovial fluid thin film, as well as retain it. And particularly for synovial fluid is a very complex fluid. And so we worked with only, we looked at only the role of glycoproteins and glycosaminoglycans. And then also using SFA, we measured the nanomechanics of the thin film under normal load and confinement, and see what the hysteresis of the different types of synovial fluid film is. And we also can get viscoelastic properties of the thin film that is formed and measure shear compliance or the shear modulus of the thin film. And in future, we want to incorporate grafted Zwitterionic polymers to modify the load-bearing surface. So use Zwitterionic polymers in combination with the synovial fluid for better lubrication. And so some of the work that our lab we have done is mimicking the surface interactions in different types of models. So in a very simplified model with just silica or mica or a quartz crystal, synovial fluid components are physisorbed, and using a quartz crystal microbalance with dissipation, we can monitor the real time, the formation of the thin layer, as well as the retention of it. And from dissipation or the loss of energy information, we can also model the shear compliance of the thin film. And in an also oversimplified model, we can first deposit the precursor film consisting of fibronectin or collagen type 2 or a mixture of both these components and physisorb synovial fluid components in it, and then see the retention of the synovial fluid and also study the nanomechanics of the thin film. And this is the graph in the right is showing the typical trace of the experiment using QCMD. So here on one side in the left, this change in surface mass density is plotted with real time. And so we first establish a baseline with phosphate buffer saline, and then we go up to, as we are absorbing synovial fluid components, it increases the change in surface mass density, and we then perform a washing step in order to take away the loosely bound molecules from the thin film. So here are some results from that study. We used the Sorber equation for rigid films to convert the changes in frequency to surface mass density of hydrated adsorbed mass. And if we plot that as a function of bulk concentration of synovial fluid, then we get a Langmuir isotherm, a Langmuir isotherm. And so we see that at low concentration of the synovial fluid, there is partially formed thin film, and which is more rigid. And then as we increase the bulk concentration from which the synovial fluid film is formed, there is now a loosely or weakly bound synovial fluid film that can be easily washed by flowing PBS during the washing step. And we can also estimate the dissipation, changes in dissipation from the QCMD experiments, and we can measure the JF prime, which is the frequency dependent compliance of the thin film. And the inverse of this is the shear modulus of the thin film. And we identified two different regions where for the partially formed films, it is less rigid. And as we form a fully formed film with an outer layer of loosely bound molecules, then the compliance increases, so it becomes more elastic. So and we can also measure the nanomechanics of the synovial fluid by using the SFA, and we want to compare the different components of the synovial, the role of the different components of synovial fluid. And so we compared the fully formed synovial fluid layer with hyaluronidase treated synovial fluid to cleave the hyaluronic acid, and trypsin treated synovial fluid to cleave the glycoproteins, such as lubricin. And the forces curve, typical representative curve is shown here. So the red curve is the full synovial fluid, and and the solid data points are the in compression, and the open symbols represent the decompression or the out curves. And we see that for both full synovial fluid and the trypsin treated synovial fluid shown here in blue, there is no hysteresis. However, there's a shorter compression range for the trypsin treated film. But we see a significant hysteresis when the films are treated with hyaluronidase. So this means that there is loss of energy and the irreversible molecular rearrangement because of the cleavage of the hyaluronic acid. So these experiments helped us to figure out that there are different role of the synovial fluid components that changes the integrity of the synovial fluid that is formed at articular cartilage. And for example, upon cleavage of the protein, the elasticity of the film is reduced, and the cleaving of the glycans resulted in irreversible molecular rearrangement. And we also saw that the retention of the thin film, as well as retention of the thin film from the synovial fluid dependent on bulk concentration of it. And there is a loosely bound outer layer that can be easily washed from the washing step. And so this helped us to form future work where we want to look at not only just fibronectin and collagen type 2, but a mixture in order to better replicate the precursor films that is present at the articular cartilage surface, and then absorb synovial fluid and measure the interaction forces, as well as look at the viscoelastic properties. In future, we also want to see the thin film compliance and coefficient of friction of the synovial films that are formed on combination of grafted Zwitterionic polymers. So with that, I would like to thank my PhD supervisor, Deborah Leckband, and all my lab mates, and my current research group, and my mentor, Roberto and Gracelyn Egbibulis. And I would like to acknowledge all the funding sources, and again, want to thank the DYSS organizers and chemical engineering department for having me here today. Thank you. I'd like to take any questions.
So how much synovial fluid are you testing? Like microliters, milliliters?
Microliters, it's 3 to 5 microliters.
And
That's what we have found to be enough to fill up the chamber.
yeah. And how much did depends
In the thin layer. In the thin layer, it's also in the microliter range.
So you
I
can't
have
just
to
like
go.
take out all the fluid from someone's knees to test it.
That would be great if we could do that. But right now we are just using bovine.
So I have a question about when you're using a ratio to measure, so you're trying to know the surface area that each polymer is taking up, you're using the molecular weight to calculate that.
No, I'm using the molecular weight to determine RF, the fluoriradius of the polymer, which is the size of the polymer. And so I made some assumption, that assumption being that there is 10% chain grafting efficiency. And we know that what is the maximum backing factor of the bromine terminator initiator. So using this assumption, I determined the surface density. So this way I did not need to know the area, anything about the area. It would be really great if we could measure that separately. I think that would be more accurate. Yes, so we get the thin film thickness from the ellipsometry measurements, and I think I have the formula here. Oh. Yes, so H is the thickness of the thin film that we get from ellipsometry, and everything else is known. And so we get the N, which is the degree of polymerization, and we just use the fluoriradius, estimated the fluoriradius from it. But yes, we made a lot of assumptions, sure.
Correct me if I'm wrong, the MWC model you use is mostly steric.
Yes.
Right? Do you know if there is, I know that you're using a completely control, right? Zwitterionic, but do you see any deviations potentially from the MWC model that could imply that the fact that this isn't a uncharged polymer actually plays a role in the?
Yes, so at the shorter range interactions cannot be well explained by MWC. So that's one way of knowing, like we cannot fit the entire profile. We just use these theories to get a quantitative value, so we can say, okay, this is what we are seeing qualitatively, but the chain extension is such and such, and it does increase. But we did not use it for predicting what the swelling amount would be.
And do you know if there's any reason to think that the parameters that come out of the MWC model, right, the geometric models, right,
Yes.
Would be reasonably similar to if you have repeated the same experiment for polyethylene oxide or something that would be expected to actually fit fairly well? Or do you think that, because obviously the shape is similar,
right,
and you're clearly able, but then the second question to that would be, are the polymer parameters that you're pulling out also reasonable, given that, right, they might not necessarily have been, the model has not necessarily been designed, right, for these very specific types of polymers that you're trying to use it for.
Yes, that's a good question. So we don't know the nature of the interactions that is happening. There is, well, to my knowledge, the theory don't describe anything particular to the charged side chains. So I don't really know how the parameters are being affected.
I would imagine that it would be than a typical random oil from something like, but I don't know.
Yes, especially with high salt concentration.
Yeah, so then if you're putting out things, like, I don't remember how many geometrical parameters.
Yes, it's just, yes, L naught and D. Sorry.
I don't remember how they're trying to measure the of the polymer. Very different, right?
Right.
Yeah.
So yes, so this is for the neutral brushes, there is no consideration. So this, the parameter that we're pulling out really for quantitative reasons, but they don't necessarily tell us what the actual swelling is. That's something that we can directly read off from the graphs in the experimental results, but yes. Yes, yes.
So now in your second part, your good, I noticed that you mentioned, I think that you called it that when did you go to
Yes, absolutely.
But it also didn't compress as far as the other. You know the origin of that? Like, would I interpret that as something else being on that surface? Or yeah, the blue curve, right? Like you see the black and the red.
Yes.
The different compression forms.
Right.
Spaces are not created, but they both go through the same compression.
Yes.
And the blue one doesn't.
Yes. So one of the reason why I think this is happening is because with trypsin is not it's not just cleaving the glycoproteins we want. It's probably cleaving all the serine proteins and other constituents. So we would like to investigate this further. We would like to have just lubricin specific enzyme and then compare. Then we would probably know why this is happening.
But from a SFA experimental, should I interpret that as something there on the surface?
Yes.
Absorbed on
Yes.
the blue surface? That's just not moving.
Yes.
Like that. Okay. Very cool. I have that.
Yes.
Well, no more questions for the round there. Again for joining us. Thank you for listening back. It's all good for you. You like the topic.
Thank you so much.
Please join us for the patio for yourself. All there. Syeda, that's great. Thanks for coming to visit us.
Yeah.